Hello, my name's Alan Payne and I'm speaking from England today, and I'd like to introduce Dr. Mahesh Kott, who is in Chile. And this, just to make it more complicated, the meeting is being hosted in the United States, so plenty of opportunity for international meeting, and maybe some of you are also in other countries as well. The title of today's talk is Lipid Recovery from Oleaginous Yeast Perspectives and Challenges for Industrial Applications. Some of you may have seen Mahesh's previous webinar about the oil that is being found in yeasts that have been harvested from the Chilean temperate rainforest. These yeasts are responsible for breaking down some of the material in wood as it rots. Dr. Mahesh Balwant Kott is a postdoctoral scientist at the Biotechnology Center of the University of Concepcion in Chile, and he's currently pursuing research on yeast-derived lipids for biodiesel application in a project funded by the government of Chile. Mahesh obtained his PhD in microbiology in 2016 from Shivaji University in Kolhapur, India, and did postdoctoral research at the Biofuels Division of CSIR Indian Institute of Petroleum in Dehradun, India. He's an expert in the lipids of yeast and filamentous fungi and their conversion into biofuels. And other areas of research interests include mycology, fermentation technology, and downstream processing, the solid state fermentation, bioprocess development, biorefining, refinery of lignocellulosic biomass, microscopy, biostatistics, and scientific writing. He has had more than 10 research publications in peer-reviewed journals, books, chapters, and patent applications. Now, if you've got any questions, please put them in the question box, but these questions will be addressed at the end of the presentation. And there will also be the opportunity, if you like, maybe you could indicate that in your question, if you wish, to be unmuted so you can actually join the discussion verbally rather than just doing it by writing. Again, thank you to all of you for joining us, and with that I'll hand over to Dr. Kott. Thank you.
Thanks, Alan and AOCS for having me again. As Alan has told you about my current research and what I'm going to talk today is different aspect on yeast lipids. I'm going to talk on the downstream processing, which actually involves the recovery, as I call it, instead of extraction, of lipids from the yeast biomass, and what are the challenges and future directions if you want to use these yeast lipids for industrial applications. Basically, it's going to be a review of yeast lipid recovery methods and how the yeast cell wall chemistry affects the whole process and is the key player in the yeast lipid based biofuel production process. First of all, let's brush up our knowledge about oleaginous yeast. Basically, these are nothing but powerhouses of biosynthesized lipids. By the term oleaginous means these microbes have ability to store at least 20% of their biomass, dry biomass, as lipids. The lipid accumulation may reach as high as 70%. As on the screen, you can see different genera. Actually, this ability to lipid accumulate is not widely spread. It is limited to specific genera. And again, it is species specific. Again, within the species, only few strains can be oleaginous, other strains cannot be oleaginous. Now, the interesting part about yeast lipids is that they are synthesized within the cell and stored in specialized compartments known as lipid bodies or lipid particles or lipid droplets, which are containing basically a lipid core, which is surrounded by a phospholipid monolayer similar to a cellular membrane surrounding the cell wall, surrounding the cell just before the cell wall. So in short, or in summary, lipids are intracellular and they are protected by a lipid body, which is again enclosed by a tough cell wall. That I want to stress out. In the images here on the slide, you can see some fluorescence microscopy and confocal microscopy images of yeast cells. In one of the slides, you can see the green fluorescing droplets within the yeast cells. These are nothing but the lipid droplets within the cell. And one of the image has been magnified, and the red color dot corresponds to lipid droplet. Is a very big. It is occupying the almost the whole volume of the cell. In this microscopy, we have used fluorescent probe. That fluorescent probe attaches specifically to the lipid within the cell. It stains only the lipid. The rest part of the cell remains unstained. Now, if you look at the type of the lipids stored by yeast, there are thousands of varieties of lipids which are present in normal biological world. They show a great diversity in structure, but majorly yeast lipids are made up of 80 to 90% of neutral lipids recognized by triacyl or diacyl glycerols. Apart from this, you may encounter free fatty acids, sterile esters, carotenoids, if these are red yeast or red pigmented yeast such as rhodotorula, rhodosporidium. They also have carotenoids which are classified as lipids. Apart from that, we have phospholipids, glycolipids. So basically two types of lipids: membrane lipids, these are phospholipids, glycolipids, and the storage lipids, which could be triacylglycerols, diacylglycerols. However, the lipid composition changes with respect to the carbon source which you use to cultivate yeast, as well as the temperature. Even it depends on the growth stage of the yeast. For example, if it reaches a stationary phase, the neutral lipid content decreases, and the storage lipid, the storage lipid as a neutral lipid increases, and a membrane lipid goes on decreasing mode. Even if you change the carbon source, if for example, if you grow the yeast on waste cooking oil or hydrophobic substrates, you will find more free fatty acids within the lipid body inside the cell. If you have glucose, simple sugars, you will have more neutral lipids within the cell. Now, why there is so fuss about oleaginous yeast in lipid biotechnology nowadays? Because they are emerging candidates for bio-based economy, which is defined by biofuels and bio-based oleochemicals. Now, oleaginous yeast provide a renewable lipid for all types of oleochemicals, including biofuels. Now, biofuels could be hydrotreated drop-in grade fuels such as biojet or green diesel or renewable diesel, or they could be simple transistorified products such as biodiesel. Among oleochemicals, you can prepare adhesives, you can prepare biolubricants, and there are reports on the application of yeast lipids for biolubricants and other oleochemicals, including biojet. Now, why these oleaginous yeast lipids are gaining so much interest? The reasons are clear. They're easy to scale up. They have a faster growth rate compared to other oleaginous microorganisms. They have short generation cycles. The biomass production is independent of space, light, or climate variations. Most importantly, they are amenable to genetic engineering approaches because genetic tools are easily available for yeast. So if you want to improve the lipid productivity, the lipid production, you can easily do by metabolic engineering or other types of biotechnological modifications. So basically, this is from my lab. This is just to show you that which are the different steps involved in a normal biofuel production. This exemplifies the production of biodiesel, which I've already discussed in my earlier webinar. Now, it's important to stress the fact that lipid recovery or extraction from oleaginous yeast is a difficult task, and it needs attention freshly. Just because oleaginous yeasts are good candidates, we need to focus on the different unit operations involved in using oleaginous yeast for biofuels or oleochemicals. The different unit operations are basically can be categorized as three: the fermentation, fermenting medium preparation, cultivation medium preparation, then the actual fermentation process, and the downstream processing. Now, I'm going to talk about downstream processing, that is the lipid extraction. Why it is difficult? Yeast are considered as one of the most toughest cells in the biological world because of a rigid and robust cell wall. They can tolerate organic solvents and pressure to a very great extent. I mean, there are reports of yeast tolerating pressure up to 300 megapascal. So basically, they're tough to break because of the cell wall. Cell wall plays an important role. Apart from that, when the yeast cell is broken to extract lipids, sometimes the cell extracts might contain lipases, which will again degrade the extracted lipids. There is the possibility of co-extraction of other lipophilic compounds such as carotenoids or aromatic molecules, which are also get extracted together with solvents, and they make the lipid purification difficult. And the fact which you know that the intracellular location of lipids in a special compartment, that is lipid bodies, that makes again the accessibility of lipids difficult. First there is cell wall, and then there is a lipid body, which is again protected by a phospholipid monolayer. So all these factors become challenges to obtain the optimal recovery, as I call it. Instead of extraction, I call it as recovery, because you never achieve 100% extraction. Whatever is present in the cell, you cannot achieve 100%. So we are basically recovering what is present within the cell. And these factors poses challenges for the efficient lipid recovery. For understanding this, now we know that yeast cell wall plays an important role. Now it is important to know, to give insights into the yeast cell wall chemistry, why this cell wall is tougher. Here I'm showing diagrammatically the differences between the cell wall of lipid accumulating yeast and non-lipid accumulating or normal yeast. For example, Saccharomyces cerevisiae, which is the model for normal genetic engineering in the whole yeast biotechnology based on Saccharomyces cerevisiae. So which is basically non-oleaginous, and oleaginous, for example, we can take Yarovia lipolytica, which is a model for oleaginous yeast. So basically I'm presenting lipid accumulating and non-lipid accumulating yeast cell wall here. Cell wall chemistry tells us that that chitin and mannan are present in very high quantities in oleaginous species. Now, what are chitin and mannans? Chitin and mannan are the biopolymers made up of glucosamines and mannose, which constitute the outer and inner parts of cell wall. The diagram shows the location of these polymers and the content present in both lipid accumulating and non-lipid accumulating. You can see the red colored chitin biopolymers are in great quantities in oleaginous as well as mannan. is also present in high quantities, and beta glucan is present in scanty amounts in oleaginous. Now, what is the role of this chitin, mannan, beta glucan? Chitin gives rigidity or strength to the yeast cell wall. Mannan supports this role of chitin, and it is also known to be hydrophobic, the chitin polymers. So it is clear that these polymers, if present in high quantities, will offer additional resistance to the yeasts, additional resistance to the cell lysis, as opposed to the non-oleaginous species. Although a lot of information is available on Saccharomyces and other non-oleaginous cell wall composition, scanty information is available on oleaginous species cell wall chemistry. But whatever information is available, it shows that chitin and mannan content is higher, which offers additional resistance to the cell wall. So this point need to be taken into consideration. Before opening or before breaking up the yeast cell wall, so no doubt to break open the yeast cell wall is the first step in the lipid recovery process. But even before, so basic steps exist in downstream processing, that is the harvesting or collection of oleaginous yeast biomass from the fermentation broth. Even before you have yeast in hand for the cell breaker, you should be able to recover yeast cells. And it's also a challenge when you scale up the process from lab to industrial. I'm an academician, but I have to consider the scale up issues, because on one day this process is going to scale up, and so the discussion has to be in that direction always. So if you look at the recovery, so if you look at the harvesting of this biomass, these are the methods showed up on this slide. Basically, all these methods utilize the principle of differences in density between the two phases of the fermentation broth. These two phases, one phase is aqueous medium, and the other phase is oleaginous yeast, the lipid containing yeast biomass. Now, if there is a difference in density between these two phases, you can use this method. For example, centrifugation, which is the most widely popular method to harvest the yeast biomass, but it cannot be efficient at large scale, because at large scale, oleaginous there will be higher cell density culture. The oleaginous yeast density will be similar, because they will be buoyant because of the lipid bodies. And the difference between the densities of aqueous phase and the cell will go off, and so centrifugation will not able to separate. So that need to be addressed. Now there are solutions for this. You can add flocculants. What flocculants will do, they will make cell aggregates, and cell aggregates can be sedimented. So basically, sedimentation by using flocculants is a solution to recover yeast biomass. However, there is a problem with using flocculants. Some flocculants have affinity for solvents, and they may get extracted out if you use solvents at later stage for lipid extractions. The other methods like dewatering, basically dewatering is done to concentrate, is the first step to reduce the volume, processing volume. Dewatering can be done by freeze drying or heat drying. It is very efficient at lab scale, which I have personally observed, but the questions have been raised for their feasibility at large scale operations because of the high energy consumption. So it's not economically feasible, this method. Filtration can be done, but again, different types of filtrations are there, ultrafiltration, microfiltration. But if the oleaginous is producing a high quantity of surfactant, they are known to produce surfactants. For example, Yarovia, if it is grown on waste cooking oil or hydrophobic substrates, it naturally produces surfactants to grow on this. And these surfactants may get adhere to the membranes used for to retain the phase. aqueous phase and to separate the biomass, and they may interfere with the filtration membrane, the surfactants. So basically, each method has its pros and cons, which I wanted to discuss over here. But you can achieve the balance. Like for example, you can use flocculants with maximum cell aggregate formation capacity and less affinity for solvents. You can achieve sedimentation and recover the collection of this biomass, harvesting of this biomass. Or you can modify the culture conditions to minimize the production of surfactants, and surfactants will no longer interfere with the membranes infiltration. So this could be the solution. Now, once you have harvested the cells by using any of the methods already discussed, the next step is to break open the rigid yeast cell wall. Now, thousands of methods are available in the literature, which could be categorized as physico-mechanical, chemical methods, and enzymatic as is a special class. We should categorize a special class of cell lysis. The term used here is biomass conditioning. Basically, biomass is the collection of yeast cells, and conditioning is nothing but the we are pre-treating the yeast biomass similar to the lignocellulosic biomass which you use in biofuel production. Basically, we are giving pre-treatment to the yeast cells so as to increase the cell membrane or cell wall permeability. We are going to increase the permeability of the cell wall by any of these methods. So it leads to increased and faster extractability of all the lipids. So basically, it leads to cell lysis. The ultimate result of any of these biomass conditioning method is the rupture of the yeast cell wall. Now, this could be physico-mechanical or chemical. Chemical methods involve the use of acids, alkalis, detergents as well are used. Physico-mechanical methods involve physical cell disruption methods like bed milling, high pressure homogenization as well. Microwaves are the upcoming technique. So once you obtain the cell lysate, the lysed cells are lysed, you have the cell lysate, which is further subjected to the liquid extraction by the solvents of choice. You need to separate the cell debris, and it is necessary to give aqueous aqueous washes to the crude extract to remove the non-lipid contaminants, and at the end you will have crude lipid extracts. Basically, this is a brief overview of how the lipid extraction process looks like, whether it is lab scale or pilot or industrial scale. The principle remains the same. Now, I'm going to give you specific examples which are tabulated. I'm not going to detail, but I'm going to tell the brief overview of how the different researchers have used different techniques depicted here for different applications. Acid treatment is one of the most widely used cell pretreatment or cell lysis technique. You can see here, researchers have used it for different applications ranging from biodiesel, biolubricant, even for food applications like cocoa butter, like lipid production process involved acid hydrolysis with hydrochloric acid of the cells of the different yeast strains, which are shown over here. So basically, if you see hydrochloric acid mediated hydrolysis at mild temperatures around 80 degrees Celsius for 1 to 2 hours has been done, which is followed by solvent extraction. After acid treatment, other popular cell conditioning methods are physical cell disruption methods. These are more popular for pilot and industrial scale. Now, these involve two techniques: physical shear and liquid shear based methods. The liquid shear based method is high pressure homogenization. Basically, in high pressure homogenization, what is done that fermented broth is directly subjected is directly subjected to high pressure homogenization. It is passed through a homogenizer, continuous passages there under pressure, and cells are ruptured. So and the temperature is maintained as well because of the heat level nature of the lipids, and you need to maintain the temperature. Apart from liquid shear, there is a physical shear-based method, that is bead beating. Basically, the suspension of yeast cells is added with abrasives. These abrasives, which I have tabulated here, these abrasives could range from beads, could range from different materials of beads, such as glass beads, ceramic, or zirconium steel beads are also used. Basically, yeast cell suspension is added with these abrasives, which could be beads, and it is agitated at high speed, at low temperatures. You can see the different specifications of different processes depicted here. For example, 30 minutes of bead milling is done at 4,000 RPM in one example, followed by solvent extraction for Yarrow lipolytica. For the other strains, different specifications exist. Like some people researchers have used 7 cycles, each of 42 seconds, with 0.5 gram of beads. Other people have used different specifications. Now, in one of my earlier research, I have combined glass bead abrasive method with liquid nitrogen method. So basically, there is combinatorial approach. First, cells are subjected with glass beads, and then I have added liquid nitrogen to freeze, and then I allowed the cell mass to thaw. So basically, this is cryopulverization, which I term it. So sometimes, I will come to that point later. Sometimes one single preconditioning method is not sufficient. You have to use it in combination with other method. For example, over here is liquid nitrogen I have used for freezing and thawing. So it enhances the lipid extraction. So different yeast species, different specifications, even the size of beads matters here. Depending, although the yeast cells don't vary much in size, size of glass beads have been known to play an important role in bringing out the efficient cell lysis. As I pointed out earlier, microwave-assisted extraction is one of the upcoming technique for the cell conditioning or pretreatment. Basically, it uses the energy of microwaves to heat up the solvent in contact with the sample, and it partitions the solvent. It partitions the analyte samples in the sample into the solvent. It combines both traditional solvent extraction and microwave-assisted extraction, but it all depends on the dielectric susceptibility of both the solvent and the sample. So that is the reason why polar solvents such as water or methanol or DMSO, that is dimethyl oxide, are suitable for microwave extraction, not chloroform methanol, which are the most popular lab scale organic solvents. They are not suitable for microwaves because they have very poor dielectric constants. So the beauty of microwave is that it can be applied to the wet yeast biomass. It can be carried out in presence of water. One thing I forgot to tell that yeast cells are either dried by heat drying method after the recovery, or they can be subjected to cell conditioning as it is without the removal of water. Dewatering is not necessary for some methods. For example, microwave, dewatering is not necessary because it can be carried out in the presence of water. And this wet and dry yeast biomass plays a very important role in the overall process economics, because dewatering or heat drying consumes a lot of energy, and that has been shown to play a great role in commercialization or the feasibility studies for such processes. So it is always better to have a cell conditioning method that can work with wet yeast biomass instead of dry. No doubt, with the dry, lipid extraction is more easier. These are the specific examples of microwave acid extraction. Now, these are the examples where transesterification is done along with extraction combined method. If you can, if you will look at the common factor in all these methods, the time taken for microwave acid extraction. That is, for example, one method has taken only 30 seconds. The another method has taken 120 seconds, which is opposed to the normal oxalate extraction, which at least takes 4 to 5 hours for the same amount of sample. I agree these are lab scale reports, but microwave is known to be one of the most rapid methods. It can bring extraction within fraction of second. However, another microwave might have some disadvantage. They might cause the degradation of triacylglycerols or diacylglycerols to free fatty acids. It has been observed in some cases. So you need to optimize the whole process of microwave. Another method of cell conditioning is actually the method of producing biodiesel or transesterified products. It combines cell disruption, lipid extraction, and transesterification. So it is nothing but the direct one-step transformation of cellular lipids into fatty acid alkyl esters. Previously, we have shown this method to be beneficial for fungal biodiesel production. Biomass, alcohol, and catalyst are put in one reactor and they're allowed to carry out the reaction, and at the end you will get the biodiesel. Catalyst could be acids, which are preferred because they can carry out, they can tackle free fatty acids also if they are present by esterification of them, or they can also do transesterification of TAG. So if the yeast oil extracted yeast is having free fatty acids, you cannot use alkali catalyst because it can bring about the saponifications. So in that case, acid catalyst is preferred. Now, these are the examples of in situ transesterification. I have tabulated over here. All this information you will find in my recently published article, the details of which will be given at the end of this presentation. The purpose of showing this table is that both wet and dried types of yeast biomass have been used, and mostly acid catalysts have been preferred for the transesterification procedure. Now, till now, we have seen the most popular, widely used methods. Apart from this, research fraternity is always coming up with unconventional, novel methods, which includes enzymes, recombinant enzymes, glucomannan coming from other yeast have been used to treat the yeast cell biomass and to make it more amenable to extraction. Apart from that, detergents are also being used. Supercritical carbon dioxide extraction is being developed. Basically, these are environmentally benign methods. They do less harm to the environment, and at the same time, they're highly efficient. Some techniques use green solvents. Another interesting solvent category is switchable hydrophilicity solvents. They are being used. You have to just bubble up the yeast cell suspension with carbon dioxide, and the solvent will switch from hydrophobic to hydrophilic, and vice versa. So apart from that, ionic liquids are also interesting category being used for extraction. The issues with these techniques could be scale up and the cost. But these things need to be come up for the better recovery. to put up all these methods on one platform, we need to see the pros, cons, and how much solvent is going to be used in each method, and which one of this method is having scalar potential. This chart tries to show that in one platform. Principles, of course, principle of each method is different. For example, acid works by breaking down the cell wall polymers. That brings the cell lysis. Microwave breaks hydrogen bonds in water molecule and changes the cell permeability, and it allows the solvent entry. Ultrasound works on the cavitation basis. So this is a theoretical part, but this principle need to be understood. Advantages and limitations. For example, ultrasound and microwaves are no doubt rapid, highly efficient, but the disadvantage is that you need specialized reactor. For microwave, you need a microwave reactor, and this is energy consuming process. Ultrasound as well, you need equipment to generate ultrasound waves, and ultrasound is very costly to operate ultrasound reactor. Scalar potential, definitely it need to be explored for these methods. Bead milling and acid hydrolysis are also having a disadvantage of high solvent consumption, but scale up is possible because they are easy to set up. Operational cost is not much, and bead milling has been proven successful at pilot scale operations earlier in 80s for cocoa butter fat production from yeast. Enzymatic method, the same problem is, the advantage is that it is one of the most safest method for environment. However, yeast cell wall, as I told you, is made up of different different polymers, and so to degrade yeast cell wall, you need to have not a single enzyme but a cocktail of enzymes which will act specifically on different cell wall polymers to bring about the lysis. So the cost is the issue. Enzyme cocktail, the production of enzymes for use in cell lysis is a issue, and it's also time-consuming process. In situ transcription, it is no doubt faster process. It has reduced energy requirements. The problem is with the high solvent consumption, and as I told you, it converts the whole cellular lipids into fatty acids. Impurities will also get extracted, so lipid purification is an issue. Soxalate, the age-old method of extraction, is one of the most easiest, simplest method to set up. However, it has drawbacks of thermal degradation of lipids because it's it takes a long time for soxalate extraction, and due to the time consumption, the energy consumption is also high. And it cannot, and lab scale, I mean, it also depends on the sample quantity. It cannot process, if you're doing, if you're downscaling methods, solvent, soxalate extraction cannot be of any help because it is unable to process smaller sample volumes. Now, let's have a look at the extraction solvents. I'm not going in detail. However, I would like to tell you here that there is the choice of organic solvent is very important, whether it's lab scale method or industrial scale. It also depends on the intended use of the yeast lipids, or which is known as single cell oil. If it is for food grade, you have to use, if your food application, you need to use food grade solvents. But if it is for biodiesel or biojet, you need to worry about the grade of the solvents. Another thing you need to take into account is the interaction of lipids with the other biomolecules. Lipids are strongly associated with the proteins and polysaccharides, and you should choose solvent. For this, you should need to know the cell wall composition. So here the cell wall plays an important role. If you know the cell wall composition, you know the associations of lipids with other biomolecules, and if you know the associations, you need to break the associations, the linkages of lipids with the proteins by using special category of solvents. You have to use both polar and non-polar depending on what type of lipids are present. If like dissolves like. So that is a rule. So you cannot use non-polar lipids, so non-polar solvents to dissolve non-polar lipids. For that matter, methanol is always used. A polar lipid, methanol is always included. It is miscible with water. It can work with wet yeast biomass. So if you have, there are also other alcohols like N-butanol, which is partially miscible, and extract the lipid without the aid of other organic solvents. So basically, these are not extraction solvents, but they help in disrupting the association of lipid with other biomolecules. Because here we are talking about the yeast cell. The lipids are not alone; they are with other biomolecules. So you need to get rid of other biomolecules. So for that, cell wall chemist, for that, knowledge of cell wall chemistry is very important. Chloroform is the most infamous organic solvent. So there's always a research going on to replace chloroform. One of the most lucrative option is using MTB, methyl tertiary butyl ether. As compared to chloroform, it is not carcinogenic, it is non-corrosive, it is non-toxic, so it is environmentally benign. It is safe for the personnel and the workers working on the extraction process, downstream processing, and it also chemically stable. It doesn't affect extracted lipids. MTB has been shown to be able to recover all the major lipid classes. It is easy to automate the process, so the labor is reduced, and it has been used for high throughput lipid profiling of oleaginous yeast. Another important thing of MTB is that MTB, when used along with methanol, it forms a upper organic phase as opposed to the chloroform. When chloroform mixed with methanol, the lipid extract goes in the lower phase, organic phase, and it is difficult to access. So if lipid is present in upper phase with MTB and methanol, it is easy to handle, and there are no dripping losses. This is particularly useful when you are developing or standardizing the lipid extraction process at lab scale. Well, I'll skip this. These are the some analytical methods, which are age-old methods, which are used for not only for yeast cells. These are also used for algal lipids, Falch methods, and the bland diet method. Now, the question will ultimately come: which cell conditioning method is the best? We have seen comparative, we have seen a brief overview of different techniques, methods for cell treatment, and now which technique is the best and which is suitable for scale up and which has been used for pilot or industrial scale. Right from the 80s, we have reports like cocoa butter equivalent fat production process was commercialized way back in New Zealand using the yeast Apiotrichum, which is cryptococcus, which has been reclassified as cryptococcus species nowadays. That was a pilot scale plant, and downstream was done using continuous ball mill. So basically, bead milling was used here for rupturing the yeast cells, followed by extraction using isopropanol and hexane of the lipids. In recently, the two reports are coming, which are pilot plants, which are currently showing that acid lysis is the best method, the most efficient method for recovery of the yeast lipids. It has been found to be scalable. Economic modeling, it has been integrated with economic modeling, and it has been shown to be feasible for both the recovery and the lipid yield. So to conclude, I would like to say you should know the yeast cell wall chemistry. It is very important before you will choose the cell extraction. If you choose the cell lysis method, or before you start optimizing the downstream on the lipid recovery. why you want to know the yeast cell wall chemistry, because it serves three purposes. You will know what type of polymers are present, if it is having high chitin or mannan. So it will, or what type of associations are present between lipids and other biopolymers. And depending on this, you can choose a solvent, and to also determine your cell lysis strategy. For example, if you have more tougher cell wall, you need to change the method from the mild one to a severe one. The fact is, one method, for example, for one yeast A, is there an yeast B, is there one method which has shown optimal yield for yeast A cannot give the same yield or same recovery for the other yeast, because there are differences. Now, why these differences exist? Why the same method cannot be applicable for all? There is no gold standard in yeast lipid method. So underlining this fact, you have to test different methods for strain of your interest, the yeast of your interest, and you need to assess different methods. And to assess different methods, the yeast cell wall composition, the knowledge of yeast cell wall composition will help definitely to design this. Another thing is that if the use of defatted yeast biomass, which is also a factor need to be taken into consideration. There has been market for beta glucans. Beta glucans, as I told you, they are present in yeast cell wall. Now, once the lipid is extracted, the yeast cells, the cell debris is left. Now, it is reaching beta glucans, and beta glucan have been shown as a good food additive, and it has found applications in industry. So it is of commercial interest. So if you know, cell wall composition will also tell us this, how much beta glucan is there and whether it is feasible to use this method or not. So to conclude or to show the way ahead, the future direction, here is a biotechnological approach which I proposed in my earlier research, that you can actually play with the yeast cell wall. You can actually modify the yeast cell wall. This is called as cell wall engineering using the genetic tools available. Now, you are aware that yeast cell wall is made up of three biopolymers, mainly chitin, glucan, and mannan, along with proteins. Now, if you have enzymes degrading these different polymers present in the yeast cell wall, they will definitely they will definitely bring about the cell lysis. Now, this can be done by expressing the genes coding for different enzymes from different sources in the cell. It can genes can be cloned. Signal peptide will carry this. These are the genetic engineering technology which will include signal peptide. Signal peptide will direct the proteins, that is mannanases, chitinases, or glucanases, which are the enzymes or proteins which are expressed in the cell wall, and they will be present in the cell wall. However, what signal peptide will do, signal peptide will direct these cell wall enzymes to secrete outside the cell wall, in the vicinity of the cell, and they will bring about the digestion or the cell wall degradation. So basically, you will be engineering yeast with enhanced autodigestibility. So this will ultimately reduce the use of solvents or enzymes, because cell is autodigestible. We have made it autodigestible, so it will ultimately reduce the use of solvents, and you can use more milder methods, and lipid can be extracted out. So this is a proposed approach of yeast cell wall modification, and this approach has been used in plant cell walls. Plant cell walls have been modified by expressing cell wall modifying enzymes such as glucanases, xylanases, and they have been made more amenable, more susceptible to the cell lysis. The details of whatever information you have seen in this presentation can be found in my previously published article recently. You can reach me on LinkedIn as well as ResearchGate and other profiles. I'm open to collaborations or consultancy for any matter. And finally, I would like to acknowledge my funding agency, that is Fondesit from the government of Chile. I'm grateful to the University of Concepcion, and last but not the least, AOCS for having me again and giving me opportunity to share whatever knowledge I have with all of you. Thank you.
Thanks very much, Mahesh. Let me see if I can camera on. I think, can you say? Yes, here I am now again. Yes. We've had a couple of questions put in the question box. I don't know whether you can see them or not. First of all, there's a question from Esteban Lopez, who says, could you please explain a generic process diagram to recover oil from these yeasts? So I think what he's asking, you know, is there actually a little schematic that you have that shows, you know, how a practical process might work? And Esteban, if you'd like to be unmuted and follow up on Mahesh's answer, then just let us know.
Okay, so what I understood from this question is this diagram, probably. Yeah. I need to explain this generic process diagram. Okay. Basically, what is done here is the first step, you have the fermentation broth or the suspension of yeast cells. The first step is to harvest or to collect the yeast cells from the biomass, because it is having water, nutrients, everything. You need to concentrate. Now this harvesting or collecting the yeast can be done by different techniques, which I have explained here. You have centrifugation, you can use filtration, or you can simply dewater. Dewatering is also done prior to centrifugation or ultrafiltration to basically to concentrate the yeast cells. This can be done by freeze drying, heat drying. And once you have the cells, now this diagram takes up. Here, once you have the cells, you can use different techniques depending on the scale of the process, depending on the availability, the feasibility, the energy input, everything, even the intended application. For example, you can use acids, alkalis, or detergents. You can use microwaves. Basically, these are the cell lysis methods. We can call it as cell mass conditioning. The conditioning of the cells is very necessary. This conditioning is necessary because each cell wall is rigid and robust. You need to condition it. You need to increase its permeability. You need to make it more susceptible to the lysis. These methods will ultimately result in full or partial cell lysis or the rupture. Rupture is nothing but once a cell wall is broken, the internal contents are coming out. Now these internal contents have come out, and you can see here I have shown diagrammatically the cell lysate. Now the cell lysate have cell debris, other cell wall, other cell constituents like proteins, carbohydrates. Each cell has everything. Now this is the aqueous cell lysate. You need to extract the lipid out of it. You're still not done. So you need to, then the normal liquid extraction process comes into picture. Basically, you are adding solvents. After adding solvent, will extract out the lipids. And now you need to get rid of cell debris. It is separated. This is where the beta-glucan thing, which I've explained, comes into picture. It can have commercial value, cell debris. Once you have the crude lipid extract, you can wash it with the wash solution. It is recommended because lipids have tendency to form micelles and reverse micelles, and these reverse micelles trap water-soluble non-lipid impurities. So you need to get rid of. For this, the crude lipid extract need to be washed with wash solutions, and then you get the crude lipid extract. So this is how this is generic, very generic process diagram to recover oil. I hope I answered this question. Alan?
Yes. What I'll do is I'm just. Can I do that? No, I think Amy, can you unmute Esteban for a moment? So if he wants to add any follow up with that.
Yes, just give me 1 moment to find him in the attendee list, and I can do that.
Yes, I tried to unmute him myself, but I don't seem to have the power. It's only you have the power, it seems.
Actually, you did unmute him. He is self-muted.
Oh, did I? Oh, I did unmute him.
You did it just right, Alan. But he has to now unmute himself as well.
Oh, I see. So I don't know whether I've unmuted him or muted him again now.
Okay, I'll check. But everyone's, can't normally unmute themselves, but you've given him permission. Nope, you have him open. So if he chooses to unmute himself, and all you have to do is click the microphone next to your name, and it will turn from red to green, and then you should now be able to speak.
Okay, well, if Esteban hasn't got any follow, there is another question that we can follow up with if Esteban is satisfied with that answer. This is a question from Rick Tyner. It says Saccharomyces slurry is a common waste stream from the beverage industry. Now, Saccharomyces is actually a form of yeast, of course. It's the yeast used for making beer or whatever, wine and all that sort of stuff. Is there any chance that this waste could be used as a source of useful oleochemicals, or does the limited oleo content prevent that? So here's what he's asking: is all the yeast formed by fermentation in wine and beer and all that stuff? Could you use it as a source of oleochemicals itself?
It's a very good question, and this strategy has been used earlier, but not for the application which has been mentioned here. The application mentioned here is for the useful oleochemicals. It has not been used source of useful oleochemicals, because to have oleochemicals you should have some oil content. So Saccharomyces is a non-oleaginous, so obviously it will not be having any oil to be used for any oleochemical production. But the Saccharomyces slurry, which is coming from the beverage industry, is a good source, could be a good source of nutrients and vitamins and growth factors, and these can be used for formulating the fermentation medium for the next batch for the oleaginous yeast. So basically it has been used, recirculated. The circular economy is upcoming technology where the waste products from one stream becomes the input for the next stream. So this Saccharomyces slurry is a good source of proteins, B vitamins, growth factors for the cultivation of other yeast, and it has been done. Even the cell slurry from the oleaginous yeast itself has been, after the defatting, the defatted yeast biomass has been reused in the next batch. So definitely it has application, and that is I think that is the right thing to do if you want to improve the process economics, and with minimal waste generation, zero waste generation, biorefinery. So it fits in all those criteria. Thank you for this question.
Yeah. Rick has replied here in the question box, fascinating, thanks. Oh, that's good. I assume you're happy with that answer then, Rick. Something that just occurred to me is I suppose there's an incredibly large number of yeasts either discovered or maybe yet to be discovered, which could yet find applications in these in these things.
Yes, definitely. So in microbiology, I'm a microbiologist, so we have this terminology for this. We call it as bioprospecting. So what Alan, you are talking about is bioprospecting, and it need to be taken at large scale. As we have discussed earlier, there might be some other yeast present in the Chilean rainforest waiting for to get explored. So bioprospecting is what is needed to find to discover yeast with more interesting lipid composition, with more interesting properties. So yeah, because, for example, in the north of Chile, we have extreme environments like Atacama Desert, world's driest desert, and there have been reports of exceptional biodiversity of microorganisms reported from that part of the Chile and on the South Antarctica, because temperature is known to affect the lipid composition. I remember I worked on the same yeast, but isolated from different environments. One yeast we had from tropical waters near the refinery, near the Indian Ocean, and one yeast was isolated, the same genus, the same species, isolated from Scottish cold waters. Both had totally different lipid composition just because of their habitat from which they have isolated. If you will genetically map, they will look the same, but they have, just because they have been isolated from different environments, made the difference. The bottom line is that the lipid composition is very sensitive. It is affected by a mere shift in temperature, or if you change the food for the yeast, if you will feed the yeast, if you change the food of the yeast from glucose to oil, the lipid will change. So definitely there is a lot to be done in this matter, right from the discovery of new yeast up to the understanding the yeast cell wall chemistry, because it is the purpose of this webinar is to stress the fact of yeast cell wall is an important aspect which was neglected. It needs to be in the limelight because it opens lots of avenues. Like one question has been asked for Saccharomyces slurry. So defatted yeast cell debris is one of the issues need to be tackled. And if you know the cell wall composition, you can apply it. If you know it is having a high beta glucan, you can get more benefit or output by diverting the beta glucan, extracting beta glucan from the cell debris. So it can make it more economical.
Well, thanks very much, Mahesh. I don't see any more questions at the moment, and I think we're just about out of time. So, thank you all for attending. Thank you for your questions. Thanks, Mahesh. It's a very interesting. I find it's a very interesting topic. What else may be lurking in the Chilean rainforest that we can make use of? I don't know. Amy, do you want to say anything more at this stage?
No, just thank you, both of you, for collaborating on this webinar. It was fantastic. And thank you, everyone, for joining us. And let's definitely do this all again.
Yeah, I look forward to that.
Thank you for having.
Thank you. Thanks, everyone, and good night or good afternoon, wherever you happen to be.
Yeah.